Bismillahirrahmanirrahim. Today we are going to discuss about inflammation. In our previous lecture we discussed about the definition, introduction, types and an overview of the acute and chronic inflammation. This is the series of online lectures for the third year students, and today in inflammation we will discuss leukocyte activation, their effector functions and outcomes of acute inflammation. We make a brief review of our previous lecture first, and in our previous lecture we discussed various steps that occurs during inflammation, and we discussed that after the initial trigger, which is caused by two things. Remember that the two initial triggers for the inflammation are the external triggers, that may be something from the outside like microorganisms or tissue injury in the form of physical, thermal or mechanical trauma, or due to other reasons that come from the outside. And the second thing, that is the internal trigger, that is the necrotic and dead tissue. So the two things that trigger inflammation are the external triggers and the internal triggers. When the body is attacked by an external trigger, or if there is dead tissue which initiate inflammation in the form of an internal trigger, what happens that initially, after the brief and transient vasoconstriction in the area of inflammation, there is vascular dilation. And the purpose of vascular dilation is to bring the leukocytes, the forces or the military of the immune system of the body, named the leukocytes, that are brought to the site of inflammation and injury. So the vascular changes are two. Number one is the vascular dilation, in which the vessels are dilated and there is increased blood flow to the area, which results in two cardinal signs of inflammation. One is the warmth, and the second sign is the erythema, or increased redness due to increased blood flow. So the warmth and redness, the two cardinal signs, they are caused by vasodilation or increased blood flow to the site of the inflammation, or where the damaged tissue is located. And this is called the accumulation of leukocytes, or the flow of the leukocytes to the damaged area or to the area where the dead tissue is lying, is known as the leukocyte recruitment. Now, how leukocytes are recruited? Normally, in the laminar flow or in the axial flow, the leukocytes, they flow in the central zone of the blood flow. But when tissue injury occurs, or if there is a dead or necrotic tissue lying, what happens, that these leukocytes migrate from the central flowing zone to the peripheral zone, and they get attached to the endothelium and again get detached. This is called rolling or loose attachment of the leukocytes. By loose attachment we mean that they are attached and then detached, and this is called the rolling. This rolling of the leukocytes on the endothelium is mediated by mediators known as the selectins. After this loose attachment, another molecules come into play, and they are known as the integrins. And the integrins cause firm attachment of the leukocytes to the endothelial wall. Now, once they have firmly attached to the wall of the endothelium, the next step is the migration of the leukocytes from the blood vessel into the extravascular space. And we discussed in our previous lecture that the interendothelial gaps that occurs due to various factors like endothelial contraction, direct endothelial injury. What happens that these leukocytes, they leave the blood, they leave the vasculature and migrate into the site outside the blood vessel where the dead tissue or the invading microorganism is lying. Now, these molecules that cause loose attachment and firm attachment of the leukocytes to the vessel walls, and those that cause the leukocytes to leave the circulation through the movements known as the diapedesis. For example, as in M.B.B.S Students you study in parasitology, that moves. So in that way, the leukocytes migrate from the blood vessel into the extravascular space. Now, these molecules are stimulated by various cytokines known as the inflammatory mediators. And these various cytokines are involved in the expression of selectins and integrin molecules on the endothelium. For example, they are stimulated by the tumor necrotic factor and interleukin 1. So these cytokines, they increase the loose attachment, firm attachment and migration of the leukocytes from the blood vessel into the extravascular space. Here another molecules come into play, and they are also small cytokines, and they are known as the chemokines. And these chemokines attract the leukocytes toward the invading pathogen or to the site where the dead tissue is lying. And they are known as chemokines because they promote the directional migration of leukocytes towards the site of the tissue injury and towards the site where the pathogen is lying. Now, these cytokines are released by tissue macrophages and other cells, including endothelial cells and leukocytes. And all these cells, they respond to the presence of a pathogen or the presence of dead tissue. And the leukocytes, they are migrated to the site from the blood, flowing blood from the blood vessels. They come out and they are then activated before they fight against the injury. In the initial phases of inflammation, particularly in acute inflammation, the predominant cells that come into play and fight against the invading agent are the neutrophils. But within 24 to 48 hours, the neutrophils, they are replaced by chronic inflammatory cells. For example, lymphocytes and macrophages, and they are more stronger than neutrophils to fight against the invading agent. Now, here we come to study activation of leukocytes. The leukocytes that are recruited to the site of tissue injury and to the site where the dead tissue is lying, they are activated before they are able to fight against the invading agent and the dead tissue. And as we discussed that the microbes and other initiating inflammatory initiators that induce inflammation, they have certain patterns and they present these patterns, and these patterns are recognized by the leukocytes. This is important that the leukocyte must first recognize some components of the microorganisms, and these components are known as the PAMs, pathogen associated molecular patterns, and they constitute the external factors. Now, coming to the dead or necrotic tissue, they also present certain components which are recognized by the inflammatory cells, and they are known as the DAMs, damage associated molecular patterns, and they constitute the internal factors. Along with the PAMs and DAMs, certain inflammatory mediators released by the inflammatory cells also stimulate the leukocytes, and the leukocytes recognize these PAMs and DAMs along with certain inflammatory mediators through their receptors that are lying in their surface, and these receptors are known as the pathogen recognition receptors. Now, once the leukocyte, the inflammatory cell, recognize the PAM and DAM through its pathogen recognition receptors lying in its surface, this causes activation of the recruited leukocytes. Now, what are the major recruited leukocytes? We divide them into granulocytes, those inflammatory cells that contain neutrophils, for example, neutrophils, eosinophils, basophils, and mast cells, and the agranulocytes, those cells that do not have granules in their cytoplasm, and these are the lymphocytes. Normally, the monocytes, which are mononuclear cells, they circulate in the blood. But when tissue injury occurs, if the tissue is invaded by a pathogen, these monocytes, they migrate from the blood flow to the extravascular space, and they are here changed into to more stronger cells, which are known as the macrophages and dendritic cells. The macrophages and dendritic cells are also known as the antigen presenting cells because they will present certain parts of the pathogen they do ingest to other inflammatory cells, and in this way they will amplify the inflammatory reaction. Now, in the first step, the vascular changes that occur in the form of vasodilation and in the form of increased permeability through which the leukocytes leave the blood and migrate into the site of inflammation. Now, the second step is the activation of leukocytes. First, leukocytes were recruited, and then these recruited leukocytes are activated. Now, as we know that the microbes and products of necrotic cells and inflammatory mediators, they are recognized by the pathogen recognition receptors, right? And this recognition then leads to the activation of leukocytes. By what we mean that the leukocytes are prepared to fight against the invading pathogen and the dead tissue which is lying there. So leukocytes, once they are activated, now what they are supposed to do? They will phagocytose the particle, the pathogen, the dead tissue, and then after phagocytosis, they will cause destruction of the phagocytosed microorganisms and dead tissue by the major component of reactive oxygen species. The reactive oxygen species are used here in the leukocytes to kill and digest the invading organisms. During the cell injury, we discussed the reactive species in the form of free radicals, and I told you that these free radicals are normally produced during certain physiologic metabolic processes, but the body has mechanisms to scavenge these reactive species, because these free radicals can cause, potentially they can cause severe injury to the healthy tissue. And in many clinical diseases, these reactive species, they are involved in the pathogenesis of so many diseases, and they are also the targets of the modern medical therapies, because these reactive species that cause damage to the healthy tissues. For example, in hepatitis, which is chronic, acute and chronic inflammations that occur in the liver, in other inflammatory diseases, and in so many autoimmune diseases, these free radicals are reactive oxygen species. They are very important, and most of the medicines that we use, we target these reactive oxygen species. In addition to their intracellular killing and degradation of the pathogens, the leukocytes, they kill and destroy the microbes that lie outside the cells, which we say extracellular microbes. And for this purpose, the leukocytes use their DNA and form extracellular traps, nets-like structures that entrap the pathogens, and in this way the pathogens are killed. Now, the activated leukocytes are also supposed to produce various mediators that include arachidonic acid metabolites and other various cytokines, and these are involved to amplify the inflammatory reaction by recruiting and activating more and more leukocytes. Now, what is phagocytosis? In phagocytosis, as I told you, that what happens, that the first step in phagocytosis is the recognition and then attachment of the particle to the leukocyte. Here, number one, you can see there is recognition and attachment of the microbe that bind to the phagocyte receptor in a vacuole, in a small vesicle, which is known as the phagosome. Now, the cytoplasm of the leukocytes also contain vesicles known as the lysosomes, and as we know that inside these lysosomes there are hydrolytic enzymes. And what happens next, that the phagosome, which is formed and containing the microbe, it fuses with the lysosome, and once it fuses with the lysosome, now the combined vesicle which contains the lysosomal enzymes as well as the pathogen is known as the phagolysosome. Now, the next step is the destruction of the pathogen within the phagolysosome, and this destruction is mainly caused by the production of the free radicals known as the reactive species, and these reactive species are used to degrade and digest and kill the pathogen, right? Now, so in addition to these free radicals, the leukocytes also use certain enzymes also to kill the microorganisms. Now, let us talk about a little about the free radicals. In cell injury, we discussed free radicals, and I told you that the free radical, what is a free radical? It is a molecule with an outer orbital containing unpaired electron, and it is highly unstable, and it will readily combine with other healthy molecules and will damage the phospholipid layers, the nucleoproteins, and after it attacks another molecule, it converts the molecule into another free radical, and in this way these free radicals, they induce a catalytic type of reaction converting normal phospholipids and normal components of a cell into reactive species, and in this way an amplifying catalytic reaction is initiated that will cause serious injury to the healthy tissues. But here in phagocytosis, this reaction, or the reactive species or free radicals, they play a beneficial role, and they are used, these free radicals are used to kill the ingested microbes. Physiologically, during oxidative phosphorylation, one electron is added to oxygen and it is converted into a free radical which is known as the superoxide. Then another electron is added to it and it is converted into hydrogen peroxide. Now hydrogen peroxide is converted into another free radical known as the hydroxyl ion, and the final outcome of this reaction is the water. This is physiological, but the conversion of the oxygen into water midway during this reaction, free radicals are produced in the form of superoxide, hydrogen peroxide, hydroxyl ion, and other free radicals. And normally in physiological processes, the free radicals that are produced, they are neutralized by certain antioxidant mechanisms, certain scavenger systems of the body, so that they don't get accumulated to cause injury to the free radicals. And what happens that if the production of the free radicals is more than the scavenging mechanisms of the body, what happens that they will cause damage to the cell membrane by causing peroxidation of the phospholipid bilayer, and they will cause damage to the nuclear DNA through their oxidation. Now here we are talking about the phagocytosis, and in phagocytosis these free radicals are used by the leukocytes to kill the microorganisms. In the surface of the leukocytes there is the NADPH, nicotinamide adenine dinucleotide phosphate, which is oxidized by the enzyme known as the phagocyte oxidase into NADP, and during this reaction what happens that the superoxide, which is a free radical, is produced. Now this superoxide itself is a free radical, but it also can combine with the nitric oxide, which is derived from the arginine by the enzyme nitric oxide synthase, and after this nitric oxide combines with the superoxide, it forms another strong free radical known as the peroxynitrite. Also, the free radical superoxide is converted into hydrogen peroxide. Hydrogen peroxide itself is a free radical, and it is converted into hydroxyl radicals through Fenton reactions in which free iron is involved, and it is also converted by the enzyme myeloperoxidase to hypochlorite, which is itself a strong free radical. Now here we can see that we get 5 free radicals. One is the superoxide, the second is the hydrogen peroxide, the third is the hydroxyl radicals, the fourth is the hypochlorite, and the fifth is the peroxynitrite. And all these 5 free radicals, they are used by the phagocytes to kill the microorganisms. And here we can see that the free radical induced damage is having a beneficial role, which is used by the leukocytes, particularly the phagocytes, to kill the microorganisms. Right? So all these 5 free radicals that attack cell wall of the bacteria and other bacterial components, and they will kill the bacteria and will degrade them. In addition to the free radicals, the phagocytes also contain certain enzymes, for example lysozyme, lactoferrins, cathelicidin, and defensins, and through these phagocytic enzymes they make holes or perforations in the wall of the bacteria, and in this way kill it. Now here we come to the summary of the leukocyte effector mechanisms, and we can say that the leukocytes, they kill the microbes and the dead cell, the two triggers for inflammation by phagocytosis, and it is followed by their destruction in the phagolysosome through free reactive oxygen species and nitric oxide. So this destruction is caused by the free radicals, for example reactive oxygen species and nitric oxide, which are generated in the activated leukocytes, and in addition there are lysosomal enzymes that are also used to kill the bacteria and dead tissue. But here one thing that happens, and that is not good for the body, and that is that the reactive oxygen species and the enzymes which are used by the leukocytes to kill the pathogens, they are sometimes released into the extracellular environment, and here they can cause damage to the healthy tissues. And the mechanism that function to eliminate microbes and dead cells, which is the physiological role of inflammation, is also capable to damage the normal tissue. And here we come from the physiological role of inflammation to the pathological consequences of inflammation, which is destruction of the healthy and living tissue. Now, the various enzymes and lysosomal granules that are contained in the cytoplasm of the leukocytes, contained in the lysosomes, they have the potential, in addition to killing the microorganisms, they have the potential to damage the healthy tissue. And there are certain circumstances. One is known as the regurgitation during feeding. By that we mean that when the cells rupture, these lysosomal enzymes are released and potentially they can damage the healthy tissue. And the other circumstance in which pathological part of the inflammation comes into discussion is the frustrated phagocytosis. You know that there are certain microorganisms and certain other invading agents that are very hard and resist to the action of the leukocytes, and the leukocytes attempts repeatedly to kill these microorganisms and these invading agents, and during this attempt there is lot of damage to the healthy tissue, and this circumstance we say this is the frustrated phagocytosis. And there are certain diseases and certain microorganisms which are very resistant to the killing and degradative action of the leukocytes, and the repeated attacks of these immune cells on these organisms can cause substantial injury to the healthy tissue. In addition, there are certain substances which damage the membrane of the leukocytes, and these are injurious substances, and for example silica particles, for which there is no machinery with the leukocyte to kill them, and the repeated attacks of the leukocytes on these substances which can cause damage to the membrane of the leukocytes can cause substantial injury to the normal and healthy tissue. In this slide you can see that normally the reactive species are free radicals and enzymes like proteases, hydrolases, which are produced. The body has the mechanisms known as the antioxidant mechanisms, for example dismutase enzymes and other antioxidants like vitamin E and other things. But when the production of the oxidants and proteases, if it overrides the antioxidant mechanisms of the body, what happens? That there will be injury to the healthy tissues. And normally the oxidants, they should be balanced by the antioxidants, but whenever the production of the oxidants and various enzymes like proteases exceed the production of the antioxidants and antiproteases, they will cause damage to the healthy tissue. Now, this damage to the healthy tissue occurs as a part of the defense reaction by the leukocytes. For example, resistant pathogens like Mycobacterium tuberculosis or certain viruses which are difficult to eradicate and repeatedly attacked by the immune cells, potentially they will cause damage to the nearby tissue, and the nearby tissue here suffer the collateral damage. Sometimes the exacerbations of the consequences of the original injury, for example, as we see in myocardial infarction, when the tissue gets the supply of the blood is restored, this restored blood supply carries in itself lot of neutrophils, lot of reactive species and other damaging agents that cause additional injury to the tissue known as the reperfusion injury, for example in myocardial infarction, and we have studied it in the cell injury. Sometimes the immune reaction or the response of the leukocytes, it is inappropriately directed. For example, in autoimmune diseases, what happens that normally our immune cells recognize our body own tissues and they do not make a reaction against them. But there are certain conditions known as the autoimmunity or autoimmune diseases in which our immune system fail to recognize the body own systems and attack them. As we can say that normally a home pet dog will not bite the persons of the house, but if they start to biting the own person's house members, then we can say that this is an autoimmune response in which the immune cells attack and damage the body own tissues. Sometimes the inflammatory response may be exaggerated, more than needed and more than normal, and in this case we come to the hypersensitivity reactions, because there are so many antigens and allergens in the atmosphere, but everybody and each one doesn't develop the bronchial asthma, but certain individuals are there that are supposed to be genetically susceptible and they respond inappropriately to these allergens and antigens, and the person develop hypersensitivity reaction, for example as we see in bronchial asthma. Now what will be the result of an acute inflammation? And as I told you that acute inflammatory response may be caused by ischemia or infarction, it may be caused by bacterial infections, it may be caused by certain toxins and trauma in the form of physical injury or thermal injury. So what happens that the initial inflammatory response, if it is restored and the vascular changes and neutrophilic recruitment and certain inflammatory mediators, when the injury is slight, if it is limited, what will happen that after the acute inflammation is over, there will be resolution, and by resolution we mean that the tissue, the damaged tissue will be restored to its normal anatomical and physiological function and structure. And what happens in resolution, that there is clearance of the injurious stimuli, there is clearance of the mediators and acute inflammatory cells, and replacement of the injured cells, and the normal physiological and anatomical structure and function of the tissue is restored. But if the inflammatory reaction is severe, it will lead to pus formation in the form of abscess. So the acute inflammation may be resolved, or it may lead to excessive injury or pus formation, and or sometimes if the injurious agent is resistant, for example the viral infections, chronic infections like tuberculosis, and if there is persistent injury, repeated exposure of the healthy tissue to inflammatory triggers, and if the inflammatory reaction is inappropriately directed, for example as we discussed in autoimmune diseases, what will happen that acute inflammation will progress into chronic inflammation. And in chronic inflammation, what happens that there is formation of the new blood vessels known as the angiogenesis, and there is infiltration of the mononuclear cells like lymphocytes and monocytes, macrophages, and chronic inflammation doesn't go into resolution, because in chronic inflammation there is extensive tissue injury, and this extensive tissue injury will result in fibrosis and scar formation. The same will be the situation if there is excess pus formation, excess tissue injury, and the chronic inflammation and the excessive tissue injury will lead to the fibrosis, in which what happens that the damaged tissue is replaced not by the original parenchymal tissue, but it is replaced by the scar tissue, and here fibrosis occurs, and automatically the new tissue which is formed will be a scar tissue, and it will be unable to perform the functions of as that were for the original normal anatomical and physiological functions of the tissue. They cannot be restored, and there is scar formation and fibrosis. Now at the end, we come to summarize the events of acute inflammation. So first are the vascular changes, which includes the vasodilation and increase permeability. So the vasodilation will lead to the cardinal signs of erythema and warmth. Increased permeability will lead to the extravasation of the leukocytes through the interendothelial junctions into the extravascular tissue, where the invading pathogen or dead tissue is lying, and the initial force is in the form of neutrophils, and in the acute inflammation the neutrophils, they leave the blood vessel. Initially they get loose attachment to the endothelial wall, first by the loose attachment and caused by the molecules selectins, and then strong attachment caused by the molecules integrins, and they will migrate then through interendothelial junctions into the site of injury, and there are certain mediators which act as chemotoxic agents, cause chemotoxis. By chemotoxis we mean that they pull the leukocytes toward the site of the tissue injury or towards the site where the dead tissue is lying, and then the leukocytes will perform phagocytosis, in which they will kill and degrade the offending organisms, and this killing and degradation is performed by enclosing the pathogen in the phagosome, and then fusion of the phagosome with the phagolysosomes, and inside the phagolysosomes the damage to the pathogen is caused by the reactive oxygen species and free radicals, and also by certain enzymes in the form, for example, defensins, and these enzymes lie in the extracytoplasm of the involved leukocytes. And the outcome of inflammation will be either in the form of resolution, in which the invading pathogen is removed, and the exudate that is formed is restored, removed, and at the end we get the restoration of the normal tissue architecture, pathological and anatomical resolution. So, or if the invading agent is so strong to be killed by the microorganism, to be killed by the leukocytes, or if there is persistent injury, what will happen? That the acute inflammation will evolve into chronic inflammation, and in chronic inflammation there is extensive destruction of the healthy tissue and the damaged tissue, and the result will be the formation of fibrotic tissue, and this we call as the scarring.